everyone, in this video I'm going to explain two important topics in biochemistry, the ester bond and the urea cycle. So first, we'll look on how an ester bond is formed between a carboxylic acid and an alcohol through a condensation reaction. Then we'll explore the urea cycle, which is a vital metabolic pathway in our body that helps remove excess nitrogen by converting it into urea. So first, what is ester bond? So ester bond is a type of chemical bond formed when an acid, usually a carboxylic acid, reacts with an alcohol. So this picture here will show you how a ester bond forms. So when they react, the oxygen and hydrogen from the acid and a hydrogen from the alcohol join together to form water. So the remaining parts join together, making a bond called a ester bond. So that's all for the ester bond. So let's move on to the urea cycle. So urea cycle is a series of chemical reaction that takes place mainly in the liver. So its main job is to remove excess nitrogen from the body by converting it into urea, which is then excreted in the urine. So this part here is the liver. So the liver is the main site of the urea cycle. So as you can see at the picture, the amino acid metabolism produces free ammonia, and this ammonia is toxic to the body and it cannot be excreted properly by the body. Let's say that the right part of the cell is cytoplasm and the left side is where the mitochondria. So cytoplasm is where most of the steps occur and where urea is produced, while mitochondria is where the cycle begins. So the urea cycle actually begins inside the mitochondria, where carbon dioxide and ammonia combine to form carbonyl phosphate. So after the formation of carbonyl phosphate, this combined with amino acid ornithine lead to litroline. This reaction catalyzed by the enzyme ornithine transcarbamylase. So the next step to litroline transported into the cytoplasm, it combined with the amino acid aspartate and lead to the transmission of arginosuccinate. And the next step, the enzyme arginosuccinate acts upon the arginosuccinate and hydrolyze to produce two molecules, fumarate and the arginine. The last step of the urea cycle, the enzyme arginine acts on the arginine to produce urea and ornithine. The ornithine is utilized back into the urea cycle, whereas the urea is a soluble compound. It dissolved into the blood and it's transported to the kidney to be excreted into hemophilia. So that was how the ester bond are formed and how the urea cycle works.